So thank you everyone for joining us today for this special presentation. Our invited speaker for this month's Variant Scientist Network event is Abbye McEwen, MD, PhD. Dr. McEwen is a molecular pathologist at the Brotman Batty Institute for Precision Medicine and a senior fellow at the Center for Multiplexed Analysis of Phenotype at the University of Washington. She completed her MD PhD at Northwestern and her pathology residency and molecular genetic pathology fellowship at UW, where she spent 4 years as an attending physician on the clinical exome service. Her research focuses on bringing multiplexed assay of variant effect data into clinical variant classification. She is co-investigator on an NHGRI ROI advancing the use of functional data and clinical practice, and she leads development of MAVE MD, a clinical decision support platform that translates MAVE data into formats usable by clinical laboratories. She is also a lead author on a forthcoming paper that uses functional evidence to resolve variants of uncertain significance at scale, reclassifying roughly 3/4 of 16,000 VUS. Dr. McEwen has developed curricula on MAVE data for clinical variant classification that have been deployed both locally and internationally. Finally, she serves on the AVE ClinGen Functional Data Working Group. So let us all come together and welcome Dr. McEwen.
Right, good morning. Can everyone see my screen?
Yes.
Okay, great. And do you see the slides or the presenter view?
I believe it's the presenter view. Yes,
okay.
It is.
Is this better?
No. The presenter view is better, yeah.
Okay, perfect. All right, thank you for helping me troubleshoot that. All right, so good morning, everyone. I'm excited to be here today. Thank you for inviting me. So basically, I was asked to come and talk to you guys about our experiences using multiplexed assays of variant effect for resolving clinical VUS. And I'm structuring my talk around some frequently asked questions that you guys submitted ahead of time and that we've gotten repeatedly during our educational seminars at other places. So the goal is simple. By the end of this talk, I want you to feel comfortable picking up a MAVE data set and kind of getting an intuitive sense of how to use it. I'm here today on behalf of the AVE ClinGen Functional Data Working Group. With me today is our coordinator, Laura Muffley. I just wanted to give a shout out to her. If you have any questions about the working group, she's your go-to contact person. All right, so here's our roadmap for the next hour. So I'm going to go through 5 questions in order. What is functional data? How do you build a controlled data set? How to decide how much evidence to apply to a functional data set? What to do when assays conflict? And how to actually find the data. If you can hold questions for the end, that would be great, but do jump in if something's urgent. So what I want to do is just start with the basics. Every talk needs a grounding. So everyone here in this room is presumably a variant scientist and knows that every single variant we look at clinically gets sorted into one of these 5 big buckets: benign, likely benign, uncertain, likely pathogenic, or pathogenic. So this is based off the 2015 ACMG framework. And the idea here is that each of these big buckets maps to a probability that a variant is causal of disease. So pathogenic is above 99%, while benign is less than 0.1%. And you're noticing this big uncertain zone here in the middle. And our job as molecular pathologists, laboratory geneticists, and variant scientists is to move things out of this big VUS bucket, either toward the benign side or the pathogenic side. So here's the problem driving all of this. We've gotten a lot better at testing people over the years. NGS technology has gotten a lot cheaper and genetic testing has become a lot more routine in clinical practice. But this is a double-edged sword. As we test more and more people, we find more and more VUS. And you can see on this graph of Clinvar-Missence VUS that this is an exponentially growing problem. So this is a headache for you guys as variant scientists, for us as laboratory directors, and most of all for the patients and the clinicians wanting an answer. So we need lines of evidence that can help us resolve this problem at scale. And everyone here is aware that to move a variant, we use several different independent lines of weighted evidence, things such as the patient's phenotype, whether that variant tracks with disease in a family, how common is that variant in the overall population, whether it came up de novo, is it in case control studies or in the literature otherwise, what do variant effect predictors say, and what I'll be talking about today, data from functional assays. So the first question I want to address is one of the most basic ones, and I want to define it clearly up at the front of my talk because this gets kind of usefully used. So what exactly is functional evidence? So there are really two things here that people often conflate with functional evidence. For this talk, I'm going to be talking about functional evidence, which is at the top of this slide. So these are measurements that are derived from an assay where a specific variant is selectively altered or controlled for. And this maps to the BS3, PS3 evidence codes. Sometimes people call functional measurements derived from patient material also functional evidence, but we think this fits better under the PP4 code for phenotypic evidence. So for the rest of my talk, I'm going to be talking about functional evidence derived from model systems, not from patient material. So this figure here breaks down the two evidence types side by side, and it's worth reviewing again for a minute. On the left here is phenotype evidence. You can think of this as observational evidence for a patient. Here we use patient-derived material, whether this be blood, cultured fibroblast, bone marrow, etc. And we utilize this patient-derived material for further direct observation. And this can be things like looking at tumor histology or microsatellite instability. It could be looking at DNA methylation or bulk RNA sequencing. And in this case, you know that the patient has the molecular phenotype, but you don't specifically know that the patient's variant is what's causing that molecular phenotype. On the other hand, functional evidence is where you experimentally control for the variant. So in this example here, we have a workhorse lab cell line. The variant of interest is being introduced via a CRISPR system. And we can look at the phenotype of the cells with and without a variant. And this allows us to control for very specific variant effects. We know that the single variant that we introduced is responsible for whatever molecular or cellular phenotype we're looking at. So here are a couple more examples. You're probably familiar with biochemical genetics in the lab. We can use an alpha-galactosidase assay to assess for Fabry disease. We can look at a mitomycin C assay to look for radial chromosomes for Fanconi anemia. These are two types of data that can come from patient specimens. And here are another two examples of model systems that have been genetically controlled in this top assay. Human T RNA synthetase has been assessed in a yeast system, and on the bottom is a mouse system, which recapitulates a human phenotype. And in this system, a single variant of interest was introduced. So before MAVEs, getting functional data meant performing a traditional in vitro assay. So one had to sequence a patient who presented to clinic, get those results back, find a workhorse cell line and introduce that protein into those cells, test its function, and read out some sort of signal. And this usually happens one variant at a time, and it can be slow and laborious, and often you get your answer back months to years after your patient has presented to your clinic. And this is frustrating because these traditional assays simply do not scale. We're finding variants now at a pace faster than we can do these one-at-a-time approaches. So the gap between variants discovered and variants understood keeps widening. So you have to assay variant 1, variant 2, variant 3, and variant 4 in an individual well of a single dish, which makes this really untenable long term. So here's the question that's really changed our field. What if you could look at a whole pool of variants at a time rather than a single variant? What if you could do this for thousands of variants in a single experiment? And that's the idea behind multiplexing. So that's what multiplexed assays of variant effect, which I'm going to call MAVEs from here on, do. So you build a library of thousands of DNA variants, and you run them through an assay all at once. This assay here is a growth assay. Each different color represents a different variant. Here the variants are introduced. You put them into your workhorse cell line. And you apply some sort of pressure over time. Variants that are harmful drop out, and those that are not harmful persist. In this example, we can sort out the ones that are harmful versus not harmful by sequencing at different time points and calculating a functional score. So the end result of one of these large multiplexed assays is essentially a variant effect map. Every possible variant will get a functional score, so you can see at a glance which variants are tolerated and which break protein function. So you can think of this as basically a lookup table for protein function. And this idea isn't really niche anymore like it was maybe half a decade ago. The number of MAVEs performed for human diseases have absolutely been exploding. This map here just shows the breadth of different diseases and model systems that have been used to study human disease-related genes that span diverse disease phenotypes such as cancer, cardiovascular, metabolic, and then a potpourri of other things that we like to call rare disease. So one of the questions I get over and over again is, how do I build a control data set? So say you've got a MAVE data set in hand. Before you can use it for classification, you have to determine how much evidence to apply. And that all depends on how you benchmark and control your MAVE. So how do you build a good control data set? Before talking about building a control data set, I wanted to revisit a couple of terminology points here. I like to think of controls as both analytical controls and clinical controls. This is my pathology brain speaking here. So the analytical controls tell us, does this assay work? Is it separating normal from abnormal? And then the clinical controls are saying, how predictive are these results of being related to human disease? So you really need to do both when you're assessing MAVE's performance. So analytical performance is the first thing I'm going to touch on here. So this asks the question, is normal separated from abnormal? In this example MAVE here, we've got a functional score here on the x-axis with a variant count on the y. Deleterious variants are over here, whereas normal variants are over here. And you can see that putative benign variants, which are represented by synonymous, and putative nonsense variants, which are putative pathogenic variants represented by nonsense, are pretty well separated. And if you don't see this kind of good separation, your assay probably doesn't work very well. If you don't have a large control set of synonymous and nonsense, other things you can do are look at other variants that have been characterized by other assays. So you're more looking at the protein function or biochemical function. So over here, you'd have variants that are known to look like they're normal in your traditional assay, or variants that look like they're abnormal in your traditional assay. The other kind of control variants are the clinical controls, and this is related to clinical performance. Are the disease-causing variants separated from the non-disease-causing variants? So instead of graphing synonymous and nonsense, this time I'm graphing benign and likely benign in blue and pathogenic likely pathogenic in red. And the way most people do this is to look at variants from ClinVar, their control set. Other options would be other large databases such as HGMD, BRCA, Exchange, etc. I've seen people curate manual data sets from the literature, and I've also seen people use data sets curated from internal laboratory data. So from this example, you can see that the benign and the pathogenic are pretty well separated, meaning that the disease-causing variants are fairly well separated from the non-disease-causing variants. So the next question is, how do I even decide what to use for those clinical control variants? So thus far, there's not been a lot of guidance on this. If your gene has a lot of variants, you're lucky, and you can default to using ClinVar controls. But the CanVig group in the UK has currently been working to provide some guidance. So Claire Turnbull and Sophie Allen have kindly provided me a sneak peek at their draft, which is not out, but a preprint is coming soon. And I'm going to share with you a little bit about those details today. So these are the draft recommendations from CanVig. I'm not going to read all of them to you, but the headlines are that if you're looking at missense variants, your truth should consist of benchmarking missense variants and not synonymous and nonsense. Clinical validation should be performed by a separate lab from the people who originally did the assay. Truth sets can be modified for stringency to increase power. And there are some things we can do when we don't have enough variants to make a pathogenic or benign truth set. And that's what I'm going to be talking to you about next. Those are the questions we get most often. So I get this question asked often. What if there are not enough benign controls? I think people are historically have been more likely to put interesting things in ClinVar, so this leads to a lot more VUS and pathogenics than benigns for some genes, which leaves us with an imbalanced control set. One way around this, recommended by these CanVig guidelines, are to make a set of quasi-missense variants. And what this data set is are missense variants that aren't in any database like ClinVar yet, but if you were to take them and apply some ACMG rules to them using population cutoffs and in silico evidence, you could classify them as likely benign. So this figure from this Rowland's paper in 25 goes through this workflow. Here you would look at all possible SNVs in your gene of interest, and then you would go through systematically and exclude anything that looked like it could be pathogenic, whether that be a ClinVar classification, a case control signal, and silico predictors, or Splice AI. And once you have that data set, you can further look at it and call anything that meets BA1 criteria likely benign, and anything that meets BS1 criteria plus a benign predictor score to be in this quasi-benign truth set. So the other question we get is, what if there are not enough pathogenic controls? And this bites me often because for a lot of our cancer genes, missense is not really a well-established mechanism of disease. So there are a lot of truncating and splicing variants in ClinVar databases, but there just aren't enough bona fide pathogenic missense variants. So how do you prove that your assay is even picking up on missense as a bona fide mechanism of disease? So if you're lucky and your gene has a single mechanism, like a tumor suppressor gene, which is loss of function, which is measured by your assay, you can do something called bulk association, where you take all your variants and you pool them together, all the ones that are deleterious missense in your assay, you pull them together and then look to see if your clinical phenotype is enriched with that variant set. And I'm going to show you an example on the next slide. So this is from a paper a couple years ago that did a large saturation genome editing study of the RAD51C gene. Again, there weren't a lot of missense variants in ClinVar they could use for validation controls. So what they did was do some biobank analysis where they looked at variants with different functional classifications versus cancer diagnoses. And relevant to us is this panel at the bottom that looks at female hormone-sensitive cancers, so breast and ovarian. So if you look at all variants which were abnormal in their assay, you do see an effect. And if you look at only the fast depleted missense variants, you also see an effect. So these are the ones that are abnormal in their assay. These are all the variants that are fast depleted, including the premature truncations, and you see that the effect size is basically the same. So this helps you understand that missense variants are indeed a bona fide mechanism of disease for RAD51C. So now that I've touched a bit on how to make a truth set, once you have that truth set in hand, how do you then use that truth set to decide how much evidence to apply? So the question here is, how do you convert data into evidence? So I also want to talk about the terminology of data versus evidence here, because this is something a lot of people get hung up on, including myself. I've probably already used the wrong term throughout the talk. So data is the functional score and the functional classification. So this is the output of the assay. So in an assay, something could have a score of -2 and be functionally abnormal, or a score of 0.5 and be functionally normal. But this doesn't tell you anything about what kind of evidence to apply. So we must take that data using a control data set and convert it to evidence. And that maps to our ACMG evidence codes, such as pathogenic strong and benign strong. So to translate data into evidence, we compare distributions of our control pathogenic and benign variants and look at the overlap. So basically, this slide is just to basically give you an intuitive impression of what the strengths of evidence are without having to do any math. So for strong, you can see that there's very little overlap between the benign distribution and the pathogenic distribution. This is best case scenario. There's always going to be a couple things here that are miscalled. Either they're wrong in ClinVar, or your assay is missing a mechanism of disease. If there's more overlap, this would equate to moderates. And if there is even more overlap, we decrease the evidence strength further to supporting. But how do we quantify this? As variant scientists and laboratory directors, we're really not allowed to eyeball things. So let's talk about how to actually put a number to that with a likelihood ratio. So everyone here at some point has probably been exposed to the concept of a likelihood ratio and odds. So in this framework of classifying a variant, the prior odds are our initial guess that a variant is pathogenic. Once we have functional evidence in hand that has been converted to a likelihood ratio, we can multiply that times our prior odds. This is our new information. And then we get our posterior odds, which is basically our updated guess. And in plain terms, the likelihood ratio asks, how likely is this result normal and abnormal in variants that we know are pathogenic versus variants that we know are benign? So if abnormal shows up more in pathogenic variants, that's a high odds ratio and strong evidence. You'll also see likelihood ratios called odds path, and mathematically this is equivalent. Feel free to use either term. All right, and then I wanted to also say the difference between an LR positive and an LR negative. An LR positive is the likelihood ratio of something being pathogenic given an abnormal result. The LR negative is the likelihood ratio of something being pathogenic given a normal result. So now let's make this concrete with real numbers. We're going to calculate the likelihood ratio for both an abnormal and normal result. Let's do abnormal first. Before we even do that, if you were going to eyeball this, this looks pretty good. I would guesstimate that you would get strong just by the amount of overlap. So you can see over here in abnormal land, there are very few benign variants. And in fact, 184 of the 186 pathogenic variants do score as abnormal. So that's our numerator. And you can see that only 27 of over 1,000 variants that are known to be benign score over in the pathogenic range. So 27 over 1171 is our denominator. And if you put this together, you get a likelihood ratio of 42. And if you don't want to do the math on your own or program an Excel spreadsheet, the Enigma Consortium has published an LR calculator, which is pretty useful. I've put a link here, and I think Lara can help me put that link in the chat for everyone to have for future reference. So now that we've looked at our LR positive, we want to look at the LR negative, which is the likelihood ratio for a pathogenic variant given a normal result. So if you do this, we see that 2 out of 186 pathogenic variants are called normal, and 100 or 1,132 over 1,171 are benign. So if you divide your numerator by your denominator here, your LR negative is 0.01. And to convert,
Dr. McEwen,
yes.
Sorry to interrupt. You have a little pop-up that's at the bottom of your screen.
Oh, no.
Sometimes
Okay.
it's
Thank you.
covering your presentation. No problem.
Thank
Sorry.
you. I was looking at my other monitor. Luckily, there's nothing personal on there. All right. Now that technical difficulties have been resolved, we can think about converting this likelihood ratio to evidence strength. So this is a tool that's been provided to us by the Bayesian framework for adapting the ACMG rules. And I've turned this into a number line so you can see it. So for the benign side, the greater you are, the higher the evidence strength. Pardon me, for the pathogenic side, the higher this number, the greater the evidence strength. For the benign side, the lower this number, the greater the evidence strength. So our benign strong, our LR of 0.01 maps to benign strong, and our LR of 19.2 maps to pathogenic strong. All right, so the next question is, what do we do if assay results are conflicting? This is something that worries a lot of people, and it can feel like a crisis, but usually it isn't. So let's walk through it and think about it a bit. To understand resolving conflicts between different assays, I like to take a step back and understand what different assays are actually measuring and what they're doing. And I like to think about this as a pyramid. Up here at the top, we have all the tiny little molecular interactions or protein properties that you need for anything to happen with that protein in the cell, or for a specific function to happen within the cell. So this could be something like protein stability, interaction with other proteins, interactions with DNA, etc. And breaking any one of these things can break protein function, but it might not necessarily break all protein function or be the only way to break a protein. Down here in the next tier of our pyramid, we've got major functions of a protein. The example I used here was BRCA1 and BRCA2. So these are involved in homology-directed repair, transcription, cell cycle, and even RNA splicing. And then down here at the bottom, we have assays that are more integrative that measure all of protein function or most of it, which are things like our saturation genome editing based growth assays. So up here, we get information about mechanism. It tells you how the protein is broken, but it only tells you one way. Down here, we can detect many ways to break a protein, but you don't get any information about how it is in fact broken. So keep this in mind as we're talking about navigating conflicts. So what do you do if results really do clash? Step one is take a deep breath and make sure it's really a conflict. I'll talk about that in a second. The second option is to say I can't apply any functional data to this because they're conflicting. Step or option 3 would be to try to determine which assay is the best and use that one. So that could be the one with the most controls or the highest likelihood ratios. And the final option would be to train some sort of model or predictor to combine the data. And this can be as complicated as a machine learning classifier or as simple as a decision tree. Okay, so first thing you do when you get a variant where you have two different assay results that are conflicting is try to adjudicate the molecular mechanism. So in this example here, we have a protein measured by two different assays. One could be protein abundance. We know that a lot of missense variants cause loss of function simply by destabilizing a protein. The other mechanism, so the other assay is looking at protein enzymatic activity in this example. So you could mess up abundance and not mess up activity. So activity can be messed up by a variety of different ways. So if I have a conflict between the abundance assay and the activity assay, I'm going to call this not a conflict and move on. The other thing I like to think about when resolving conflicts is to think about how did the investigators actually get that library into the cells? So there are two major paradigms that people use when they're putting variants into cells. One is to put variants on a piece of exogenous cDNA, like a plasmid. And here, this is just the coding sequence, so there are no introns or any other regulatory elements. And then the other way people introduce variants into cells is to use endogenous genome editing. So in this case, you make basically a single nucleotide change at the variant's endogenous locus. So this retains all effects of splicing and nonsense-mediated decay. So if I have one assay that looks at endogenous gene editing and one that looks at libraries that are made of plasmids, I asked myself the question, okay, is my variant in question, does it affect splicing or not? If it affects splicing, I'm going to probably ignore this assay over here. And that pretty easily resolves that kind of conflict. So once you've ruled out the easy things, and it's a genuine multi-assay case, you can combine the assays in a structured way. I think this is a great example from the TP53 VSEP, where they took several different functional assays and combined them into a decision tree, which allows the variant scientist to apply a final evidence code based on combinations thereof. I think this is a nice template for other people to use. It's fairly simple and intuitive. But science has gotten a lot more complicated than decision trees. And when you have several MAVEs and you don't just want to pick one, you can also do something like train a classifier. And in this paper here, this was Sean Fair's original paper where he showed the power of MAVEs for reclassifying TP53, BRCA1, and MSH and P10 variants. So at the time, there was no guidance about combining TP53 assays, and he learned that of the 4 different assays that were available at the time, they didn't have a great AUC on their own. So what he did was train a classifier which outperformed the individual assays. If you want a little bit more information about combining multiplexed assays, we do have a preprint that you can refer to. And this preprint goes into more complicated ways of combining assay data, such as using different kinds of clustering algorithms. And basically, they showed when you used an individual MAVE, you could only reach moderate evidence, but when you combined all 3 together, or pardon me, all four together, you could reach strong evidence. Okay, next question is, how do I actually find this data? You've shown me that it's useful. You've shown me how to create a control data set and to calculate evidence strength, but how do I actually get this data when I'm out in the field? I'm a variant scientist and I need to find it. So I'm going to give you a tour of the MAVEDB resource and our new clinical front end called MAVE MD. So this is the MAVEDB landing page. And just a little bit of background here. This resource was originally meant for data scientists and like basic protein biochemists. It was not designed with any kind of clinical features in mind. So we've spent the past 2 years overhauling this resource in response to community feedback to make it more useful for your particular needs. So if you go to the MAVEDB website, you can see that up in the right corner on the menu, there is a button for MAVE MD. And if you click this button, it brings you to the MAVE MD landing page, which was built with our group of users in mind. And if you scroll down on that page, you can browse a bunch of data sets that we have curated with clinical metadata. Right now, we have sufficient clinical metadata for 84 different functional assays across 40 different genes. One of the key things we heard from community feedback was we need variant level searching. The original MAVEDB database supported searching by dataset or by gene, but not by individual variants. And I let our developers know that that's not really the way it works when someone's actually classifying a variant clinically. I want to go to a database and type in my variant for ease of use. So what we did was implement this kind of search functionality. So you can search with a variant-specific identifier, like an HGBS string, a ClinGen allele ID, a dbSNP RSID, or a ClinVar ID. The other thing we did was implement a fuzzy or a guided search. If you're like me, I often don't have a perfectly formatted HGBS string. So this particular search interface prompts the user for the gene of interest, whether your variant is at the P dot or the C dot level, what the position is, and what the reference and alternate alleles are. So if we do that for our variant of interest, this MSH2 variant, we can see that it is present in one assay. This is an MSH2 loss of function assay that was done in a HAP1 cell line. So if we click on the variant, we can go to what I'm going to call a variant page. And a variant page gives you a lot of information. I'm going to start with a high-level tour that goes over the entire architecture, and then I'm going to go into the important parts in detail in a minute. So if this is a little bit general, don't feel confused. So at the top of the page is the header section. So this shows you very, very basic data like the assay name, basic metadata, the functional consequence of that variant, and any kind of evidence strength for that variant. If there was more than one assay, you would see multiple assays listed up here. Underneath the header is the variant identification. Here we've got some other identifiers, and we've got a quick reference sheet for some of the key assay details that variant scientists need to know. I'm going to go over this section quite a bit of detail in a minute. Underneath that is a section that gives you the functional classification, so the score, the odds path, and the evidence strength. Underneath that is a score distribution to show you what every variant in the assay looks like. Your variant of interest is highlighted. And finally, underneath that are more details about the evidence strength and odds path calculation. Okay, so one of the most important parts of this variant page is the assay facts label here. I like to think about this as the nutrition label on the back of a cereal box to give me warnings, something maybe high fat, something may not have a lot of vitamin C, etc. So this allows you to kind of understand what the assay is doing without having to read the whole paper. So let's go through what's actually on one of these labels. At the top is author, name of the gene, date of publication in the journal, along with the gene name and HTNC symbol. Underneath that is some basic metadata so the reader can understand what the paper actually did. So this was the paper out of Jacob Kitzman's lab that looked at MSH2 loss of function in a human cell line. And in this type of experiment, variants are introduced into this cell line using plasmid cDNAs. And then a drug called 6TG is applied, which is known to select for cells that have NMR loss of function. So you can see here that from the label, the assay type is cell fitness. We're assessing DNA mismatch repair. The variant consequence detected is loss of function, and the model system is immortalized human cells. So this allows you to kind of understand what this is without having to go back and read this entire paper. Underneath that is a section that warns you of a couple of dragons which can lie in MAVEs. A lot of them are done with cDNAs, so they're not applicable to variants that alter splicing or are predicted to undergo nonsense-mediated decay. I wanted to make that painfully obvious on this nutrition label. And underneath this is basic metadata to quickly understand how good the assay is. So here we've got our odds prof normal and abnormal, followed by an ACMG style evidence code. So here are some example assay facts from different data sets. The format is standardized, which makes it easy for the curator to understand what's going on. I want to highlight a couple examples. So in this example, this is a TP53 assay. It's a cell fitness assay that looks at DNA damage response. But the key thing pointed out here is that the mechanism detected is dominant negative. And that's important because we know that TP53 can be disrupted by both dominant negative and loss of function variants. So this helps you understand what's going on when trying to adjudicate conflicts or when trying to understand what the assay is actually measuring. Here in this label here, we've got the P10 VAMP-seq or protein stability assay. So here, this molecular mechanism assessed is just the regulation of protein stability, which is only one way to break a protein. So I wanted it to be obvious that when a curator looked at this label, that this assay was only measuring one possible way to mess up a protein. Okay, after that, assay facts label is a histogram. The default view shows every single variant in the data set. Functional score is on the x-axis, variant count is on the y. The normal and abnormal intervals are highlighted in blue and red, respectively. You can see that the MSH2 variant that we searched for is highlighted with this black bar, and that it's over on the benign side. But for a variant curator, we want to know more information. We like to look at things in the context of what's going on clinically. So what you can do is switch views to clinical view and look at this same data set, but this time, in addition to shading our normal and abnormal intervals, we're also breaking things down by ClinVar classification with benign and likely benign in blue, pathogenic, likely pathogenic in red. In this example, ClinVar January 2025 is shown by default. I just wanted to point out here that the data set shown may be different than what the original authors of the paper used to benchmark their MAVE. Another thing you can explore are the different variant consequences in a data set. So this would be missense, nonsense, synonymous, etc. This data set's pretty boring. It's all missense variants. But for some of our data sets, we have synonymous and nonsense variants that we can use to help us understand how normal is separated from abnormal. You can see in this example assay that the putative normal variants here are in green, and the putative abnormal variants here, which are nonsense, are in maroon, and that there's a fairly good separation between these two. So that can help the curator get an intuitive sense of what the performance for the MAVE is. So the next question, or pardon me, the next section I want to talk about is thresholds and calibrations. So this is telling us what score intervals define each functional class and how much evidence is given to each functional class. Right now, if you click on this button here for calibrations, there are a lot of different options, including the original publication. However, we're also supporting community calibrations, including some done by our IGVF coding variant focus group, and we're working with VSNs to get the calibrations that they like in this as well. So calibration details include what the score ranges are for each interval, where those thresholds came from, and then what the evidence strength is or odds path that goes with that data set. So I want to give you a concrete example of why there might be multiple calibrations for a single MAVE. So the original paper out of Jacob Kitzman's lab drew the line between abnormal and normal at 0. And they did not calculate an odds path style evidence strength at that time. However, within a year, they published a follow-up paper where they actually used a clinical data set to benchmark and draw threshold lines that were a little bit more clinically relevant, and they did calculate an odds path at that time. All right, the next thing we've implemented is the ability to compare across multiple different assays. If we search for a variant of interest that's in multiple different assays, you can see that multiple assays come up this time. So this particular BRCA1 assay, or this particular variant, was seen in a saturation genome editing assay, as well as a couple of other assays as well. And if we click on the View Variant button, this takes us to the header, and this time it shows that there are 3 different assays. All 3 of them are self-fitness, but 2 of them were done in response to different types of drugs. And if you click on an assay, you basically get that variant page I showed you earlier, and you can click through them and compare results between them pretty easily. So the next question we often get, what do I do if the data set I need is not in MAVE-MD? So right now, our metadata curation process is pretty manual, so coverage is still growing. Ideally, we'd like to have the authors input the data set and the clinically required metadata. So if you do see a variant, or if you do see a data set that you think is useful, perhaps write to the authors and ask them to get it into MAVE-MD. We're happy to help authors with that if they run into any problems. And then the other thing we're thinking is that we should be working with VSEPs to lean on authors to get the relevant data sets into MAVE-MD. Otherwise, you can write to us with a Zulip chat that's on the MAVE-MD help page. And if you go to the help and support page, there's a link here for the Zulip chat. All right, and with that, I'd like to thank more people than I know how to thank. All the developers that made the MAVE-MD team possible, and then several people who've been instrumental in creating educational materials, such as Pankari Gupta. She's a research GC, Malvika Tajura. She's a recently graduated PhD student, and then Mandy Spurtle and Rihan Villani, who are our Australian colleagues who run the Australian MAVE Education Working Group, along with Laura Muffley for helping to coordinate the AVE ClinGen Working Group. And with that, I can take any questions.
I'm just looking through the chat here. Lara has been answering a lot of the questions in real time, so that's great. Thank you so much.
Thank you, Lara. She runs the world. She really does.
There's one question here that perhaps you could answer let's see here. For Priyanka Bajaj is asking, can MAVE datasets directly be used to inform clinicians' decision? If yes, are there any regulatory checkpoints associated with it?
So I don't think you would treat MAVE data differently than you would treat any other source of data. I think the clinician and the laboratory director needs to make the choice whether something's benign or pathogenic or VUS based on the integration of different sources of evidence, not just one evidence source. And as I understand it, I don't think there are any regulatory guidelines regarding that. And I don't think there are with things like NOMAD or predictor data either. But I guess I would caution a clinician from looking up a MAVE score and anchoring on that as the final answer.
Yeah, absolutely. It's a piece of evidence, just as, population studies, phenotype data, so on. So there's a question here from Scott Pugh. Is the clinical performance data taken from the assay other calibration publication, or is it some type of internal MAVE DB calculation? I do believe you addressed that in showing some examples.
Yeah, I can touch on that a bit. So right now, we are not doing any calibrations ourselves. Right now, that is up to the community and up to the authors of the paper, or up to VSEPs. But if there is a published calibration, we can upload that to MAVE-MD. We would like to enable calibration functionality in the future. So you could perhaps upload your control data set of interest and benchmark the MAVE against the variants that you think are the gold standard, but that's still a work in progress.
Here's a really interesting question. Do you know of any MAVEs that can be used to study missense variants in proteins that undergo post-translational modifications?
Yes. So we had a MAVE from a graduate student in our lab, Nick Pop, and he looked at trafficking of the factor IX protein. And in order for that protein to be trafficked properly, it has to undergo carboxylation. So one thing he did was measure carboxylation status with a carboxylation-specific antibody. There are probably other examples as well.
And then finally, in your slide with the Venn diagram about conflict between molecular mechanisms. So if you have a variant with abnormal protein abundance, but nearly wild type protein activity, or vice versa, would you downgrade it to moderate, for example?
That's a great question, and I think that's probably an example of a true conflict. What you could do is make a table of result in assay 1 versus result in assay 2 for all the variants in your 2 assays, and basically calculate an odds path on the combined assay result. That'd be one way to resolve it. But again, I think at this point, like, use your brain. I think I would have to know more about the particular protein, and if abnormal abundance. Yeah, that is pretty weird. You would expect there to be abnormal activity. So I think I probably would have to look at that one more carefully manually.
Yeah, I think you make a good point in your presentation about knowing the mechanism of what is breaking the protein and understanding that in a little bit more detail when you're interpreting your variants. How can you measure in some viability assay experiment loss of function and gain of function variance? Is there a recommended cutoff based on any calibration?
That is a great question. You've probably noticed that most of the examples I've talked about today, and even most of the examples in our field, are loss of function variants because they're pretty easy to interpret. So going back to thinking about loss of function and gain of function. So I think the first thing to do is ask the question, is gain of function of even a known mechanism for your gene of interest? You could call something gain of function in an assay because it was X number of standard deviations more active than wild type, but clinically that might not mean anything if gain of function is really not a mechanism of disease. And the next thing I would look at would be a control data set that had different phenotypes. So we know for a lot of genes, if there's a loss of function and a gain of function phenotype, I think a lot of the cardiomyopathy genes do this. I would calibrate on the different phenotypes and calibrate over 3 different intervals instead of 2 intervals. But you're right, it is a lot more complicated, and I think there is more guidance forthcoming about that.
Great, thank you. Well, I think at this point we will end. And thank you so much for this wonderful presentation. You really highlighted all the work you and your team are doing to, make our lives easier as variant scientists. So
we do have a survey.
You have a survey?
We do.
Oh, we're giving out a survey. Okay, great.
I think Lara can post that in the chat. Sorry, I didn't mean to cut you off.
No worries. It's okay.
Yeah, I wanted to make sure I got feedback from this group. Since we're making this tool for you, and if it's not useful to you, we're not doing our job. So that survey is going to ask you questions about MAVE-MD, and then also questions about what the functional data working group can do to make better educational opportunities.
Wonderful. Thank you so much for all you do. It's really great. So I wanted to let everyone know that we will be on a bit of a vacation break for the next 2 months, and we will be returning to you with more events in September. So keep an eye on that. And I wish everyone a wonderful summer. I'm going to give a